Hi everyone and welcome to chapter 16 part 4 gene control. Now before we go into chapter outline like we do usually, we are going to go and define what gene control is first. And before that, let's actually define what gene expression is first. So gene expression is something you've learned already in chapter 6. It's the process where gene is being transcribed into mRNA, which is then translated into protein. Now, gene to mRNA to protein, again, is gene expression. But the control, the regulation of gene expression is called, well, gene control or gene regulation. Same thing, okay? Gene control or gene regulation, the same thing. It's basically the process that enables gene expression in specific cells in specific time. In other words, it is to ensure that the correct gene is expressed in the correct cell, in the correct time, and in the correct context. So you need to have all of that right in order for the cell to function. Now, some genes are switched on all the time, okay, or are transcribed all the time, and these genes produce constitutive proteins. So constitutive proteins are produced all the time, but some genes can be switched on and switched off. They're only switched on when their protein products are required. Now, this is a good thing and it's very important as well because, number one, this ensures that there's no wastage of energy and resources. Okay, so when you have too much of the particular protein, it will stop being produced. Okay, or only when you need it, it will be produced. If not, it will not be produced. So it's good, save energy. Number two, you need to understand that a lot of proteins in the body do different functions, and sometimes those functions overlap or interfere with one another. So let's say if I want the cell to break down more sugar, I will make sure that the enzymes produced, the protein produced, are the ones involved in breaking down sugar, not forming sugar, right? So the protein products, aka enzymes or anything needed, has to be the ones that actually perform the cell function. If it's not required, it should switch off. With gene regulation in place, with our cell having certain control mechanisms to cater to different functions, this ensures that no expression of protein will interfere with the current cell function. I hope that makes sense. So this is gene control or gene regulation. Now, in this particular chapter 16 part 4, we are going to be learning structural versus regulatory genes and what the difference is. We're going to learn about repressible, inducible enzymes. Gene control is different in prokaryotes and eukaryotes, so we're going to mainly talk about prokaryotes here though, because honestly, it is a much simple model. Eukaryotes are much more complicated, so we learn less of that. But we do learn one example of eukaryotic gene control, which is gibberellin, which breaks down DELLA protein repressors. And this is something we've already learned before, and it's just a little recap. Yay! So this is a plant hormone, by the way, if you don't remember. So let's start off now with understanding what structural genes are and what regulatory genes are. So structural genes and regulatory genes are both genes, by the way, and don't forget the definition of genes: a specific DNA sequence that codes for a protein. Structural genes are genes which code for proteins needed for cell structure or function. Like enzymes, like all the normal proteins you think of, like actin, like myosin, like rRNA. These are stuff that needs to be coded for by structural genes. But when we talk about regulatory genes, we are referring to genes that code for also proteins, but proteins that control expression and transcription. So a gene that can produce a protein which influences other genes. These are regulatory genes, yeah? So these regulatory proteins that controls gene expression or transcription are called transcription factors. Now, transcription factors, what they do is they bind to a specific DNA sequence and are able to control the transcription of that particular gene. Now, there are two types here. You can see here already. One, repressors. Number two, activators. Activators increases gene expression, whereas repressors reduce or inhibit gene expression. Just to be clear, repressors and activators are both transcription factors, but they just function differently. They also bind to different sites. Activators are often bind to specific DNA sequences near the promoter, okay, whereas repressors bind to a specific DNA sequence called the operator. Okay, so repressor, which will become very important later, binds to operator. We don't talk about activators that much in this particular syllabus. Okay, so repressor binds to the operator. Again, it is a specific DNA sequence. The operator is also a specific DNA sequence. Okay, so we have defined structural and regulatory genes. Now let's define one more thing: repressible inducible enzymes. Okay, so what does it mean when we say that the repressor, that the enzyme is repressible or and inducible? So when we say repressible and inducible, okay, they mean different things. So what does repressible mean? Let's define that first. Repressible enzymes are enzymes which synthesis can be prevented when a repressor protein binds to an operator, a specific sequence of DNA. So repressible enzymes is not a repressor, just so you know. It just can be affected by the repressor. It just can be affected by the repressor. It is not a repressor. So if there is a repressor, it stops the binding of RNA polymerase, it stops transcription, and therefore it has the repressible enzymes is not produced. What do we mean by we say it is an inducible enzyme? Now, these are not mutually exclusive, means you can be both repressible and inducible. Now, inducible enzyme means that the synthesis can occur when the substrate is present. Okay, an inducer, it could be the substrate itself, can bind to the regulatory protein, in this case maybe a repressor, and cause the gene to be switched on. So you can see here that inducer binds regulatory protein. This is essentially a repressor and inhibits it from binding to the operator, and therefore transcription occurs. So when we say an enzyme is repressible and inducible, we really mean that it can be inhibited and it can be switched on when the substrate is present. Now, what do we mean when the substrate is present? We are going to be learning a very interesting example later, which has to do with the lac enzymes in a moment. But before we go into the lac enzymes and the details of that, we are going to talk about the general gene control in prokaryotes, which is via this thing called operons. So, operons, what are they? First of all, they're only found in prokaryotic DNA. So bacteria, archaea, okay? Prokaryotic DNA, operons make up a unit of gene expression. What is it? It consists of one or more structural genes, okay, under the control of single promoter. I'll explain it soon. We have a diagram later. And it also has a control region before those structural genes, which consists of promoter, where the RNA polymerase binds, and operator, where the repressor binds. There's also a regulatory gene that is close to the operon. Okay, let's look at the picture here. You can see the regulatory gene here is close to the operon. This is the operon right here. And you can see that it has one or more structural genes. So it can have multiple genes side by side. So these will form 4 different proteins later on. It can be cut. Okay, this mRNA can be can produce 4 proteins. And this operon here also have what we call control regions of DNA called the promoter and operator. Now again, just a brief recap on what we just talked about, maybe it'll make it clearer as well. Promoter is where the RNA polymerase binds, whereas the operator is usually where the repressor protein binds. So this is called an operon. Now the operon that we are going to learn is called the lac operon. This is one of the many operons found in prokaryotic DNA and is a very famous example and one that is important for your syllabus. Now the lac operon looks like this, okay, very similar to this, right? This is the general structure. This is a specific example. There is a promoter, there's a regulatory gene here, it has its own promoter. Okay, again, promoter is where the RNA polymerase binds. And then there's a lac operon nearby, also has its own promoter, has its operator where the repressor binds, and a lac Z, Y, lac Y, and lac A. Okay, so these are actually names you gotta remember. Lac Z, lac Y, lac A. Don't ask me why it's like random alphabets. I don't know. Okay, the scientists named it like that. Just memorized it. Anyways, lac operon is found in E. coli mainly, and E. coli is always very studied. It lives on sugar, i.e. glucose and lactose, and this particular lac operon is in charge of breaking down lactose. Now the lac operon has a set of 3 genes next to each other. Okay, again, lac Z, lac Y, lac A, and these codes for 3 repressible and inducible enzymes, and we're going to learn about how it's going to be repressed and induced shortly. Let's sort of see what the 3 different genes code for here. So lac Z actually codes for beta galactosidase, which is actually an enzyme that breaks down lactose into galactose and glucose so that the bacteria can process it better. Now, lac Y codes for permease, which increases lactose uptake into the cell. Of course, obviously, the lactose will be outside the cell at first. It needs some sort of transport protein to get in. And lac A codes for transacetylase. Honestly, I've searched online and looked at a few books, and it's not quite clear what the function is exactly. So let's just leave it like that. It just codes for this enzyme called transacetylase. So these 3 different enzymes, again, they are responsible for the breaking down of glucose, the digestion of glucose in the bacteria. Now, again, they are repressible and inducible, means they can be switched off and switched on in the presence of a substrate. So how does this occur? Now, a lot of this has to do with the regulatory gene, which is nearby, and the regulatory gene is called lac I. Okay, think of inhibitor perhaps, but lac I actually codes for a repressor. Now, this repressor is a constitutive protein. Unlike just now, the enzymes, the enzymes were repressible, inducible enzymes, which means, again, they can switch off and switch on when the substrate is present, but the repressor is produced all the time. The repressor protein is an allosteric protein and has 2 binding sites. Now, the DNA binding site is here. This is the DNA binding site here. And this here is the lactose binding site. So there are 2 binding sites, that's why it's called allosteric protein. And what happens is when lactose binds to its site, the shape of the protein changes and the binding site is closed. So here you can see it binds, and you can see the DNA cannot bind anymore because it has changed shape. And therefore, the repressor is inactivated. Now, lactose here acts as an inducer. So in the presence of lactose, which is the substrate, it acts as an inducer. It binds to the repressor, and the repressor changes shape and doesn't bind to DNA anymore. Now, what consequence does this have? Now, we need to analyze what happens when there is no lactose and when there is lactose. So what happens when there is no lactose? Again, the repressor is a constitutive protein. The regulatory gene will code for the repressor. It's called lac I here. And again, it's a name, you need to know this. Now, lac I is transcribed into mRNA, translated into a protein, which is a repressor, and the repressor, as you can see, binds to the operator. Now, the repressor is active as it is not bound to lactose. Now, RNA polymerase can bind to the promoter, but it cannot move to the operator and to the structural genes. There's even lac Y and lac A here, but it cannot do this. It cannot move. So no transcription occurs and the genes are switched off. Now this makes sense because when there is no lactose, why would you want to produce stuff that digests lactose? What happens when there is lactose? Now lactose, when it's present, is taken up by bacteria. Lactose then acts as an inducer, as we mentioned just now, and binds to a repressor. Now again, repressor is produced all the time, but now since lactose is present, it acts as a repressor, it acts as an inducer. The repressor is inactivated. It changes shape. It cannot bind to the operator region of DNA anymore. Okay, and therefore RNA polymerase can bind to the promoter and move through the operator region and through the structural genes, okay, resulting in transcription and therefore translation of the 3 different genes. So I have this like really cute animation to help you visualize things here. First, obviously with the lac I promoter, it's not very easy to play, so bear with me, no pun intended. And then obviously there's a lac promoter, a lac operon, and a lac ZAC gene. You can see here that the RNA polymerase can bind at first on both, right? And lac I gene produces a repressor. The repressor binds to that operator. So initially, you can see here that the lac ZAC gene can be expressed, but now with the repressor binding at the operator, the RNA polymerase cannot move to the operator and to the gene, and therefore no transcription is occurring right now. Now you also realize that the RNA polymerase tends to like to bind and then move around and then come off. Even the inhibitors, the repressors do that. So you can see the repressor moving away here, and that is characteristic, that is normal in the body. It's not always binding tightly. It can bind and detach randomly. Now what happens when we inject some lactose into the cell? So you can see here that when lactose binds with the repressor changes shape, no longer binds with the operator, and therefore the RNA polymerase can move to the gene and transcribe it, and the lac ZAC gene can be expressed. Okay, lac Y and lac A is not shown here, but assume it's there. And the lac ZAC gene, which codes for beta-galactosidase, can then break down lactose into galactose and glucose. And glucose and galactose can be then used further for respiration and to produce energy in the cell. So I hope that is a good visual representation for you. So what are benefits to the lac operon? This mechanism actually allows bacteria to produce 3 enzymes only when lactose is available, and all 3 enzymes are actually produced in equal amounts. So this is good because you will always have that sufficient enzymes in order to carry out processes. Now, other than that, this mechanism, you actually learn the simplified version. There's actually a more complicated version, but in the more complicated version, what happens is that when there is another transcription factor involved, when there's both glucose and lactose available, bacteria would prefer glucose because obviously glucose is the monosaccharide and lactose is the disaccharide. So glucose is more easily absorbed and digested and used in order for respiration. So the bacteria actually can repress the lac operon using this additional transcription factor in order to just focus on digesting glucose only. Now, all these would result in avoiding the waste of energy and materials of the bacteria, and is very beneficial for the cell. So that is basically gene control in prokaryotes. We saw the example of the lac operon, what happens when there is lactose and what happens when there is no lactose. What about in eukaryotes? Now, eukaryotes is a bit nuts, all right? It has a genome size larger than prokaryotes, a lot larger, and they have more different transcription factors and mechanisms to regulate gene expression. Just now was a very simple mechanism. In eukaryotes, it's very complicated, and honestly, if you go into your degree, you realize that we haven't figured it all out. 10% of our genes code for transcription factors, actually, by the way. Now, what does it do? Well, it obviously binds with the promoter region on RNA polymerase. It can recruit RNA polymerase and encourage transcription. It can activate an appropriate gene in sequence and allow correct pattern of development of body regions. Now, there is we have different parts of the body, right? We have the head on the top and feet where we walk on. Now, what is the genes that are responsible to form the head, and what genes are responsible to form the feet would be completely different genes, correct? So you need a certain way to control those genes, and therefore transcription factors come to play. Determination of sex also depends a lot on transcription factors. There are different transcription factors that are present on the X and Y chromosome, which result in certain features. It allows responses to environmental stimuli, temperature, and also, very, very importantly, it helps regulate the cell cycle, growth, and apoptosis in order to ensure that your cell cycle is normal and does not result in cancer instead, and also carries out the effect in response to hormones. And this list is actually still very short. It goes on and on and on. And the general idea is this transcription factors, they regulate gene expression. They help gene regulation. And therefore, everything that's related to function and related to timing and dependence on specific timing will have a lot to do with gene control and transcription factors. Well, as we said, it is very complicated in eukaryotes. We do not learn any particular examples, fortunately, but I just want to show you this picture here to show you just how complicated it can be. Don't worry, this is not really part of your syllabus. I'm just showing you how crazy it looks like. So this is a very tiny part of the DNA. You have promoter here, you have a gene A here. You can see the RNA polymerase bind a promoter. But you know, for this the RNA polymerase to bind your promoter, you need many transcription factors, not one, not two, many different ones that recruit others. And then there are DNA control regions. Okay, we talked about operators just now, but actually there are even more regulatory regions that could be further from the gene. And sometimes the DNA needs to bend like this in order for them to come into contact, in order to regulate the expression of gene A here. Now, obviously, this is just conceptual. This is just me telling you how complicated it be. But don't worry, this is not coming out for exams. This is just an example picture to show you just how crazy it can get. There is one particular eukaryotic example though that is simple and that you have learned already, and this is just a quick recap. If you want more details, you can go back and learn about gibberellin in chapter 15, if I'm not wrong. Now, gibberellin is a hormone, it's a plant hormone, and it actually plays a role in regulating genes as well. Now, without gibberellin present, the transcription factor PIF is actually attached to the DELLA protein. The DELLA protein acts as some sort of inhibitor of transcription. Therefore, the transcription factor, well, is bound, so it cannot bind to DNA. But with gibberellin present, it binds to the receptor, and this actually causes DELLA protein destruction. Therefore, PIF, or the transcription factor here, binds to DNA because nothing's inhibiting anymore. It can bind, and therefore there is transcription that can occur here because it recruits RNA polymerase. And therefore, due to this switching on of genes, this results in growth genes. It results in the production of amylase, and this plays a role in the growth of plants as well as the germination of seeds. So again, this is a very brief recap of what we've already learned. Please go there for more information if you don't remember already. But with that, I think we are done with this chapter. This part of this chapter to do with gene control. Again, it is easier in prokaryotes than in eukaryotes. The lac operon is an important example that you need to know. And with that, we're pretty much done. See you next week. Bye!